In the hippocampus and also in the entorhinal cortex, a particular EEG rhythm called theta rhythm is very, very important for the organisation. People think for the organisation of place or firing. And so in the same study, we actually looked at EEG activity in the theta band and other bands to see how theta activity might be changed by the loss of the vestibular system. So along the y-axis in B, we're looking at the power of EEG within the 6 to 9.5 hertz range, which is the theta band. And we found a significant decrease in the power of theta in the lesioned animals compared to the control animals. There were other changes as well, but one of the most important was that not only was the power of theta reduced, but the sinusoidal nature of the theta was corrupted. So here are some traces of theta here, and you can see in the worst case, the correlation between a sinusoid and what we recorded was about 0.09. So this was heavily corrupted theta. Again, you might ask whether this is somehow related to the movement of the animals, but here we plotted the correlation between the theta EEG we recorded and a sinusoid against movement velocity, and you can see that it doesn't actually make any difference how fast the animal's moving or how slowly it's moving. The correlation to a sinusoidal theta is consistently poorer in the animals that have had bilateral vestibular lesions. So there's electrophysiological evidence that the hippocampus is not functioning normally. And this is not functioning normally, up to quite a long time point as well. And so it's not just the acute effects of vestibular loss. Even after 6 weeks, when animals have compensated to some extent for postural ataxia and so forth, the hippocampal dysfunction persists. So we went looking naturally to see what the neurochemical basis of this might be. And so one thing we did was we looked at acetylcholine receptors in the hippocampus, particularly M1 muscarinic acetylcholine receptors, because these are so important in the mediation of theta activity. And we used receptor autoradiography at 2 time points, 7 days and 30 days. And we looked in different subregions of the hippocampus, CA1, CA23, the dentate gyrus, the whole hippocampus, and even the striatum. And what we found at 30 days, but not 7 days, was a consistent decrease in the density of these M1 acetylcholine receptors, consistent with a reduced cholinergic signal that might detract from normal theta. And interestingly, although I'm not talking about it here, we found a similar decrease in the striatum. Anatomically, we started looking at whether you could see the same kinds of volume changes in the hippocampus in rats as we saw in humans with bilateral vestibular loss. And this was a study that involved animals at about 16 months after the vestibular loss. And we started by using stereology and looking at the number of neurons in different regions of the hippocampus and the volume of the hippocampus. And we found no difference in the volume of the hippocampus as we did in humans. However, we started using Golgi labeling to look at dendrites in CA1, and what we found was that the dendritic complexity of dendrites was much less in animals that had had bilateral vestibular lesions. And you can see down here, these are 2 control groups, an unoperated group and also a sham surgery group compared to a bilateral vestibular lesion group. And what this shows is that in this case, there's a decrease in dendritic complexity and a decrease in the length of dendrites. And these are particularly basal dendritic intersections shown here compared to apical ones. So there are structural changes in the rat hippocampus following bilateral vestibular loss. So I want to turn to some of our most recent studies now, where we've combined electrophysiology with some neurochemical studies involving autoradiography and also flow cytometry. One of the most important phenomena in the hippocampus is a plastic phenomenon called long-term potentiation. and there's another one called long-term depression. And since the 1970s, it's been believed that long-term potentiation, or a strengthening of synapses in the hippocampus, underlies many forms of memory that the hippocampus is involved in. And LTP has in fact been found in many other parts of the brain too, including the visual cortex. So we wanted to find out whether LTP changes in animals that have had bilateral vestibular loss. Given that we see a dysfunction of place cells and grid cells and theta rhythm and poor spatial memory performance, which correlates with poor memory performance in humans with vestibular disorders. To do this, we did this in vivo, but here's an in vitro schematic. To do this, what we did is we stimulated afferent pathways in the hippocampus, and we've done this in a couple of different studies now. You can stimulate the perforant pathway into the dentate gyrus of the hippocampus down here, or you can stimulate afferent fibres terminating in CA1. Either way, what you record is a field potential, which in its simplest form looks like this, where this upward going waveform is called the field excitatory postsynaptic potential, or EPSP, and this downward going potential is the population spike. So it's a field potential representing a lot of discharge of neurons in the hippocampus, and you can use it as an index of the degree of excitability of hippocampal neurons. And the standard way of inducing LTP is to give a high frequency tetanic train of electrical stimulation. And what people do is they look at the response you get to low frequency electrical stimulation first, then they give high frequency stimulation, and what usually happens afterwards is that this potential becomes bigger. And this is the enhancement of synaptic plasticity called long-term potentiation. So if we just look through this very quickly, here's the normal situation with a presynaptic terminal releasing glutamate. It acts on different kinds of glutamate receptors like AMPA kinase receptors, and you get this kind of field potential. Then you give the high frequency electrical stimulation. That actually opens up another kind of glutamate receptor called the N-methyl-D-aspartate, or NMDA receptor, which influxes calcium, which induces plasticity. And now when you go back to the low frequency stimulation, the response to that low frequency stimulation and glutamate release is now bigger than it was before the high frequency stimulation, and you have a potentiated field potential, i.e. long-term potentiation, which is that strengthening of the connection between neurons in the hippocampus is thought to be an important neural substrate of memory, of the encoding of memory. So the question is, does this change following the loss of vestibular input? So these studies were done in anaesthetized animals, and the degree of electrical stimulation, the frequency of it was modified so that it was possible for the degree of LTP to go up or down, because basically if you use high frequency tetanization, then you get a kind of ceiling effect in the LTP. What we found was that if you looked at the first phase of the field potential, that is here, which is the field EPSP, or excitatory postsynaptic potential, we found no difference between sham animals and bilateral vestibular lesion animals. However, if you look at the downward going population spike, what we found was a consistent increase in LTP of the population spike relative to the sham animals. So somehow the loss of vestibular function has increased the population spike of the LTP, of field potential, beyond what you get from normal LTP in a normal animal. And so if we look down here, here's the sham animal before and after high frequency stimulation. And if you look at this population spike here and here, you can see there's a modest potentiation as a result of the high frequency stimulation. But now look at the bilateral vestibular lesion animal. Now, this was before the stimulation, the tetanization, and this is afterwards. That population spike is now much, much bigger. And so what we have here is a phenomenon called ES potentiation, where if you express the population spike amplitude in relation to the EPSP, in the vestibular lesion animals, you have a much bigger population spike for the same degree of EPSP. And ES potentiation, generally in the hippocampal literature, is associated with an increase in synaptic noise, because what's happening is the same amount of dendritic excitation is resulting in a much bigger response from the cell bodies generating action potentials. And so what you have is an increase in synaptic noise due to a lack of input specificity, which is normally associated with poorer spatial memory. I realise that it might seem, because you get a bigger response, it might be associated with better memory, but in fact it's the relationship between the EPSP and the population spike, the fact that you get a much bigger response even though the EPSP is the same, that really is ES potentiation and signals an increase in synaptic noise. So if LTP has been altered by the loss of vestibular input, and I should have said it at the beginning, this is at 30 days after the vestibular lesion, so this is a chronic time point. It's not an acute time point. What is the neurochemical basis of this? So once again, we turn to receptor autoradiography in an effort to try and understand the way NMDA receptors, the particular subtype of glutamate receptor that is important in the induction of LTP, might be responsible for the ES potentiation. So we used receptor autoradiography, where you use a radioactive ligand to bind to the NMDA receptor, and then we superimposed the autoradiographs onto Brain Atlas so that we could delineate particular parts of the hippocampus. And this is what we found. It's a little complicated, but this is 7 days after the lesion, and this is 30 days. And we're looking at the whole hippocampus in the first row, the dorsal hippocampus in the second row. I should have said that a lot of place cells are concentrated in the dorsal areas of the hippocampus. We looked at the ventral hippocampus and also the hippocampal subregions. The black bars show the results from the bilateral vestibular lesion animals, and the grey bars the sham animals. And we're looking at, in each of these plots here, we're looking at the left side, the right side, and the combined results. The basic take-home message here is that 30 days after the lesions, we see a considerable increase in the number of NMDA receptors. We see this in the whole hippocampus at 7 days and 30 days. We see it in the dorsal hippocampus at both time points. We don't see it in most cases in the ventral hippocampus, which contains fewer place cells, and we see it across all the hippocampal subregions. So there is a significant upregulation of NMDA receptors. Now, that significant upregulation of NMDA receptors could account for the potentiation of the population spike when we induce LTP. And also, it would theoretically increase synaptic noise. Because the increase in potentiation that you need has to be very specific in the hippocampus to particular synapses. Because we weren't sure of this result, we decided to do this in a different way, and in the last year or so we've optimized a method for looking at receptors on neurons using flow cytometry, which is a technique that's usually used in things like cancer and immunological research. So what this is showing here is day 7 and day 30, and with flow cytometry what you do is you separate cells, and you can use different fluorescent antibodies to tag different proteins. So going from left to right, we've got NMDA plus beta tubulin. We've got NMDA minus beta tubulin, NMDA plus beta tubulin, NMDA alone, and tubulin alone. And the black bars represent the bilateral lesioned animals, and the grey bars the control. The critical thing here is if you look at beta tubulin is a protein that you can label with a fluorescent antibody that's specific for neurons. So if you use an antibody for NMDA receptors, and you combine it with an antibody with an antibody for beta tubulin, if this group here shows the cells that have NMDA receptors but are not neurons, this group are the cells that have NMDA receptors and have beta tubulin, so they're neurons. These are just cells that have NMDA receptors, and these are cells that are just neurons. So the key result here is this one here, where we look at cells that have NMDA receptors and are neurons, and you can see that there's an increase in the number of neurons that have NMDA receptors at day 7 and also at day 30. And so what this means is that there's not just an increase in the density of NMDA receptors following bilateral vestibular lesions, there's an increase in the number of cells expressing NMDA receptors, the number of neurons expressing NMDA receptors. And we have found essentially the same result in the striatum now as well. So it's likely that the neurochemical basis of the ES potentiation in LTP in animals that have had bilateral vestibular loss, which is related to poorer spatial memory through increased synaptic noise, is produced partly through a very significant upregulation of NMDA receptors in the hippocampus. So we think that the population spike part of the field potential in the hippocampus may be increased in an attempt by the brain to compensate for the loss of normal vestibular input. And this could be related to this upregulation of NMDA receptors, which could actually be triggered by a decrease in glutamate release from the entorhinal cortex after bilateral vestibular loss. And we think that rather than being adaptive and compensatory, ultimately this plasticity increases synaptic noise through a lack of input specificity due to ES potentiation. And this may be part of the mechanism, the neurobiological mechanism for the spatial memory deficits that animals and also humans exhibit after vestibular loss. So the implications of these studies are that the hippocampus undergoes many neurophysiological and neurochemical changes following vestibular loss. Input to the hippocampus arises from all vestibular receptor groups, and it's transmitted bilaterally from each ear with quite long latencies. We're finding so far that the amplitudes of the responses to stimulation of the saccule and the utricle in general are greater than the canals, but this varies by electrode, by place in the hippocampus, and there are also some left-right asymmetries. Because of the evolutionary age of the otoliths, we have a guiding hypothesis that the otoliths, especially the saccule, are particularly important for hippocampal function, just because of their evolutionary role in sensing gravity and how important that is to the brain. And recent studies that we and other people have done in animals that have a mutation that basically eliminates otolithic function suggest that these animals develop in a very abnormal way with poor spatial memory from a very early age. And lastly, I just want to thank all my collaborators on these particular studies from France, Alice Benoit, Stéphane Besnard, Bruno Pousset, Bruno Truchet, and from Oxford, Yanfeng Zhang, who was a postdoc in our lab, Go Sato, who was a postdoc in our lab, and other collaborators, including Martin Hittier, who was a PhD student in our lab, and very importantly, Yuan Zheng and Ruth Napper from the University of Otago. Thank you very much.